So again, just great to see everyone today, and let's get started with today's event. So this webinar is part of an exciting series that was created by the Plant Gene Steering Committee. It's called the Interactive Series for Plant Transformation Protocols. This is not a traditional workshop, but it's more of a conversation. And this is an interactive session, so the speakers will be taking time throughout the event for audience questions. We have 3 different sections today for you. So our speakers will be presenting one section. We'll take time for questions before moving on to the second section. And we'll have time at the end to talk about the full protocol that's been presented today. So I'd like to briefly introduce today's speakers. First is Mercy K. Azanu. If you just want to wave hi to everyone. Mercy is a PhD candidate at Iowa State University in Dr. Ken Wan's lab. Her focus is sorghum as well as tropical and temperate maize transformation. Our second speaker is Faizo Kasul. Faizo has previously worked as a plant breeder with Uganda's National Agricultural Research Organization, and he is now completing his PhD in the Wan Lab. Our last speaker is Dr. Dib Pramanik, who graduated from Kyeonsan National University in South Korea, and he is currently a postdoctoral researcher in the Wan Lab at Iowa State University. All right, so this protocol has been divided into several sections. A speaker will guide you through a few slides and then begin answering audience questions. Questions will only be taken from the Q&A box. You should see that on the bottom of your toolbar there. So please make sure you are entering your questions in the Q&A box, not the chat. I'll be moderating questions for the speakers. Please send in your questions as you think of them. And of course, any information presented today will be available on the Plant Gene website along with the recording. So with that, Mercy, I'll turn it over to you.
Okay. Thank you very much, Amy, for the introduction, and hi everyone for being present today. So I'll go ahead and share my slides from here. Okay. Please you see the screen?
It needs to be in presenter mode.
Oh, yes. Okay.
Great. That's perfect.
Okay. Okay. Sorry. Hi, thank you everyone once again. So first of all, I would like to start with an acknowledgement. We are members of the Kanwang Lab at Iowa State University, and so we would like to say a very big thank you to our major advisor, Dr. Kanwang, for her immense mentorship, and also to Dr. Gunsabli for his mentorship as well. And we would like to say a big thank you to Dr. William Gordon Kam and Ning Wang of Koteva AgriScience for their mentorship and guidance on the leaf transformation protocol. And last but not the least, to Plant Gene for this opportunity to get to meet with the community and also share of what we know on sorghum transformation. So for today's presentation, we have a few slides which we will try to use that to capture everything that we do for the leaf transformation. We have 3 major parts, of which the part 2 is the major transformation. So I'll give a brief introduction, and then the part 2, which is the leaf transformation protocol. We have the explant and Agrobacterium preparation, which is all the preparations you do before the day of transformation. And then we have the transformation day where you combine the explants and then Agrobacterium. And then the last part where we have the callus induction and morphogenic gene excision and then regeneration. And then after that, we have a part where we address some common issues that we believe other researchers may face and possible solutions to that. Okay, so a brief rundown of sorghum transformation over the years. Now, the first transformation reports for sorghum was in protoplast, and that was done by electroporation, and there was not much recovered from that experiment. And then the first successful transformation with regenerants actually was reported in 1993 by Casasito, where they used immature embryo bombardment, and then they had a transformation frequency of 0.33%. And then in 2000, there was the year 2000, there was the first successful agrobacterium-mediated transformation of sorghum using immature embryos. Now, from that early 2000s, for the past 2 decades, and for the past 2 decades, explant that has mainly been used for transformation has been immature embryo. But within that time period, there have been other explants that have been tried, such as the leaf segments, immature embryo calli, pollens, shoot apical meristem, and embryogenic calli, of which have seen various transformation frequency. The highest so far has been 46.6% when you use embryogenic calli. And then for immature embryo, a good point has like a high record of it has been a 20% transformation frequency, which was in 2018, and also the use of mature seeds. But all these protocols were done without the need of any morphogenic transcription factors. Now, in recent times, from as far back as 2016, there was a first report of the use of morphogenic transcription factors to aid sorghum transformation. And most of those early reports were done as well using immature embryos. But in the recent years, in 2023, and then last year as well from our lab, we report the use of the seedling leaf whorl explants. Now, if you compare the first table to this, you realize that most of the transformation frequency range from 0 to less than 100%. But then with the use of morphogenic transcription factors with immature embryos, they had regeneration to as far as 69.7%. And recently, with the report of seedling leaf whorl explants, there are reports which are over 100% to as high as 1,000%, as we have reported in our recent manuscript. So this is a table from the paper just showing that with the use of morphogenic transcription factors assisting in the use of seedling leaf whorl explants, you can have varying range of transformation efficiency. And most of these genotypes were actually previously not reported, some of which were already recalcitrant, but then some of which had also never been tested for sorghum transformation. So you may ask, how does the seedling leaf whorl explant compare to immature embryos? Because that has been what has been used for over the past 20 years. That has been the basic approach which has been used for sorghum transformation so far, and every sorghum line has immature embryos so far as they make seeds, right? But one thing that has persisted is the recalcitrance of some genotypes to transformation. Now, if you live in places such as North America, where we have winter periods, which means you are not able to actually have field of sorghum plants, so that means you have to look for a way to sustain your production even during the winter period. So that calls for growth chambers, which means you have to have large space to accommodate that. And also quite labor intensive in the sense that it will take quite a while for you to get enough immature embryos for your transformation. In the case of seedling leaf whorl explants, of which the first report was by Wang et al. In 2023, the seedling leaf whorl explants can be produced year-round. In the case where we have like petri dishes, you just grow them in the petri dish and then put them in a box. They require less space, and the only thing is that they require the morphogenic genes for you to be able to induce somatic embryogenesis. So how do these morphogenic genes work? In this particular protocol, we talk about baby boom and whorl, although there are many other reports of other morphogenic genes. And so the combination of when you have your tDNA, which carries both baby boom and whorl into a plant cell, they are able to induce somatic embryogenesis such that the differentiated leaf cells are able to undergo somatic embryogenesis to produce calli, which are then, after the excision of those morphogenic transcription factors, can go on to produce lots of transgenic plants for you. And so talking about the tDNA that we use, so I mentioned that because the leaf explants are differentiated tissues, you need these morphogenic genes to help them undergo somatic embryogenesis. So a typical tDNA binary vector has both whorl and baby boom being driven by their specific promoters. And then we have an inducible Cree excision, which is within that period, which is guided by 2 LOXP sites. And then we have a visual marker and then a selectable marker. So once somatic embryogenesis has been induced, you just put the explants on media, which in this case we use ABA because the RAB17 promoter induces the Cree to, under ABA condition, to excise out the 2-LOX-P site, and then in your transgenic plant you have ZS screen and then NPT2 selectable markers present over there. So the leaf infection procedure in today's presentation is divided into 3 major parts where, like I mentioned, we have the explant and agrobacterium preparation phase. Those are the things you do before the day of transformation. And then we have the transformation day, so where you harvest your explants, combine them with your agrobacterium carrying the tDNA binary vector, and then you perform the transformation. And following that, we have the callus induction, so somatic embryogenesis, ABA step, which is where you get rid of your morphogenic transcription factors, and then the regeneration step. And mostly depending on how you carry out this and then the conditions that are available to you, it can take anything between 70 to 90 days to have rooted transgenic plants, which are big enough to go to the soil. And so at this point, I would pass it over to Dr. Divyajyoti Pramanik to take the next session.
Thank you, Mercy. So the first step is, now we'll go more detail for each steps. So first step is seed sterilization. So currently we are dealing with 2 kind of seed sterilization process. One is the seeds coming from the greenhouse or from the growth chamber. So it's a nice environment, and other part is the seeds coming from the field. So for the seeds that are coming from partially sterile condition like greenhouse or growth chamber, they looks very pretty. So we can use surface sterilization by using sequential steps like ethanol and then bleaching solutions for roughly 20 minutes, and then we wash with lots of water to remove those bleaching agents, and then we place the seeds into the germination plates, as you can see here, and we can keep them in the growth chamber to grow them further. So it takes 10 to 12 days to get the seedlings to work on further. So germination media also having the ensimidol, is a anti-GA agent. So it's not only reduce the height of the plant, but also Wang et al. Reported that it's showing improve the transformation efficiency. So we are using that protocol to get our seedlings. So as I told you, the seeds are coming from field. Those are very bad. So we get hard time to get rid of all those contaminants. So we tried different ways. As you can see here, these seeds also have sometimes cracks, and we tried with the different bleaching agents, and also the modifying the incubation period for the bleaching substerilization, but we found lots of contaminants, like mostly those are fungal contaminants. So we came up with this idea now to improve our substerilization process. So we also treat those seeds with different fungicide and try to remove all those surface contaminants. So one thing we found, like the contaminants are not from the outer surface. Sometimes it's inside. So we need to remove the seed coat. And once we remove that seed coat, we can go for the surface sterilization by using ethanol and bleach, and that actually remove lots of contaminants. So we provided one video here. So how we remove the seed coat. So as you can see here, we took individual seeds, and then by using forceps, without disturbing the embryonic side or embryonic tissues, we try to carefully remove all those seed coats gently, so that without damaging the seeds. So if you damage the embryonic side, or you get some cracks, the seeds may not grow uniformly. So that also need to keep in mind. And once we remove the seed coats, we just go for the normal self-sterilization process and then incubate and place the seeds into the germination media and then keep them into the growth chamber using a shoebox. We use a shoebox that provides us sterile condition, and then we grow them in the chamber, and roughly 10 to 12 days we can get the seedlings. So for the next step is the preparing our agrobacterium. So typically we always make a master plate. So what is master plate? Master plate is a plate where we provide a minimal media with antibiotics, and then we streak them, and then we can keep the master plate for a period of 7 days or 1 week roughly. Uh, and why we are making that? Because in many uh, experiments, we have multiple experiment in the same week, and we cannot take the glycerol stock from -80 every time. So to reduce that task, we just treat them in the master plate and keep them. And then the day of transformation, before that, 1 day, we streak the working plate. So working plate is typically YP and antibiotics. So as you can see in the figure, we just take some of the colonies and then spread by using L-shape spreader or the loop, and we spread all over the plates uniformly so that we can have a nice growth in the next day. So one thing we also recommend, we should not use old working plates, so it should be always fresh. So now we reach to the end of this first section, and we can take some questions. It's over to you, Amy.
Yeah, thank you both for that, taking us through our first section. So we have a couple of questions here in the Q&A. Just a reminder to everybody to please use the Q&A box, not the chats. And then I did see some raised hands. Again, we're just going to be taking questions through the Q&A. So our first question is, have you tested other inducible promoters other than the ABA inducible for Cree? I'm assuming this is talking about the morphogenic gene.
Okay. Thank you for the question. Okay, so we have tested heat shock promoter specifically for sorghum, and we saw response of phenolic compound production for that. So at least aside the ABA treatment, we know that heat shock promoter does not really work for us in our condition as of now. So, but those are the two we have mainly tested for now.
Great, thanks. Another question was, any special reason for using AB plus Thi for the mother plate, but using YEP plus Thi for the working plate?
Yeah, so YP is a minimal, the mother plate is usually a minimal media. It grows slower. But YEP is a rich media, so it can give a quick growth. So roughly overnight, if you streak them, we can get it in the next day. And then it provide enough amount of cells to go further for your transformation, because you need a 0.5 to 0.6 OD to work for the co-cultivation.
Okay, great. Thanks. That's an important detail. Last question here, and then we'll move on to our next section, was how long do you soak the seeds so that you can easily remove the seed coat as shown in the video? After removing, you do the surface sterilization, right?
Yeah. So we usually soak with water for a couple of hours, and that makes it easier to remove the seed coat because it's having get the water inside partially. So and we can go exactly what we are doing for normal sterilization, like a couple of minutes in ethanol and then 20 minutes in bleaching solution, and then wash with lots of water, and then we can place them in the germination plates. Yeah.
Okay, great. All right, well, thank you everyone for your questions. Keep them coming. If we don't get to your question in the section, we'll have time at the end to go back and to some of these questions. All right, so with that, I will turn it back over to our speakers.
Okay, so now in this section, I'm going to take you through the Agrobacterium infection process up to the time of regeneration. So on the day of Agrobacterium infection, before you do the infection, there are a couple of things that we have to do. First and foremost, we have to do heat pretreatment for the seedlings that are 10 to 12 days. And we do that at 45 degrees Celsius and at a relative humidity between 60 to 70%. So as you can see in our lab, we use the Fisher brand incubator, which does not come with an optimized humidity system. So what we do to get the required humidity is that we use a tray, and then we put some paper towels in that tray, and then we put water, and then we know that for 1 hour or 1 hour and 20 minutes, we get the required relative humidity. So the relative humidity is important because you don't want your seedlings to dehydrate. And at the same time, heat treatment for 2 hours has been shown to improve transformation frequency by threefold. The purpose of heat treatment is that it enables better intake of Agrobacterium by inducing heat shock proteins in host cells. So after the heat treatment, what happens is that actually you get your plastic box that has your seedlings, then you take them to the laminar flow hood. But of course, we need to really be clean and a clean environment. So we kind of like use 70% ethanol to wipe our box. Then on the laminar flow hood, it is advisable that you remove one petri dish at a time and you keep others in the plastic box, because you remove them from the heat. You don't want to kind of like change conditions. Then now you begin on preparing your seedling reefers. So you begin harvesting, and how we harvest them is that actually we remove 2 or 3 outer leaves that are really old so that we can expose the young actively dividing leaf whorls. And the way we do this, I'm going to explain this in a video. So you can see in this video is that you use forceps, you remove gently the outer leaves. Using forceps, you divide the outer leaf into 2, as you can see. Then you expose the young active dividing leaf whorl. So you use a pair of scissors, as you can see, gently, then you cut off these upper leaves. Then you hold using forceps, then you cut up at the leaf base that gets into contact on the media. You harvest your leaf whorl, then you put it into 700 J. So the reason that's why you do it, because you want to maintain it healthy before you do Agrobacterium infection. So then comes another part, which is really interesting in this protocol, which is the blending part. So the blending part is important because you are going to cause wounding. Wounding kind of like generates these leaf segments that you are going to infect with Agrobacterium. But even when you're creating the leaf segments, you're wounding, but you don't want to do it so much. At the same time, you don't want to do it less. So this is a critical step. So what we do in our lab is that we use this type of blender. It's called the Quiznut blender. I'll play a video, then I'll explain. So this blender makes us to kind of like have uniform leaf segments from these seedling leaf whorls. But then also, you can also do the hand chopping. But of course, hand chopping to get 2 to 3 millimeters leaf segments varies from person to person, but it also works. So I'm going to play a video to explain what happens in blending. So in blending, in the blender, you have your 700 J solution. Then you have your leaf whorls. So you blend using the low pulse, and then you shake it a little bit so that every part of the leaf whorl comes into contact with the blade. And the pulse, we do between 5 to 10 pulses. This also depends, because when you have a new blender, you know it cuts very fast. But then when you have an old blade, also it takes longer to chop the leaf segments here. So you can see on the top here, you may have some residual leaf whorls that have not been cut well. So it's advisable to remove them. Then you use a scalpel to kind of like to cut and have a uniform required measurement of 2 to 3 millimeters. This is really important because when you overblend them, what happens is that actually you are going to cause most of the programmed cell death, other than kind of like inducing these leaf segments to kind of like to regenerate. But we say that hand cutting is also possible. You can use the scalpel and then you can hand cut them. So after that, we put them into the cell strainer here. So the reason as to why we do not do blending with Agrobacterium solution, because most of the times we work with different constructs. But if you're working with one construct, you can blend in the 700 and IO 700F that has the Agrobacterium. So after that, putting them into the cell strainer. So there are a couple of things that I have to talk about. One is the preferable size, because the preferable size, like for example, like 75% to 90% of the leaf segments that you get after blending, so they have to be between 2 to 3 millimeters. So this has been tested by our lab and then also by the Wang et al. 2023 protocol. When you have less than 2 millimeters, is that actually these ones do not give you very good color and they do not regenerate well. And this is really important because if you don't do the required measurement of the leaf segments, it impacts on your regeneration. So the other thing that I want to talk about, now that you have your leaf segments, you have your explant ready, now you can proceed with Agrobacterium infection. So Agrobacterium infection is also an important process here because the strain that we use in our lab is LB 4404 type minus. We all know that actually when you work with this strain, as long as you put it into liquid media, it tends to kind of to agitate and clamp. So when you're using this strain, it is important to really work very fast and efficiently. For example, if you're preparing your Agrobacterium solution from your now working place that you have prepared a day before, you make sure that you prepare your Agrobacterium solution. I'm talking about in the 700F solution that has acetylshiringon, breakthrough, which is a surfactant that kind of like makes the Agrobacterium adsorb onto the leaf segments very fast, between 1 to 3, maximum should be 5 minutes. You should not exceed that. Then after that, you get your leaf segments, then you put them into your Agrobacterium solution. So you're seeing you have 2 falcon tubes because you have 2 different constructs. So after dipping them into the Agrobacterium solution, so it is known that the OD that is preferable is between 0.5 to 0.6. So to increase on the tDNA integration is that actually we put our falcon tubes onto a shaker and we make them rotate them at 70 RPM. So this is also important because it slows down on the clamping of the Agrobacterium, but at the same time, it uniformly distributes the leaf segments so that they are, so the DNA integration is really better. Then after that, after 20, and you do it for 20 minutes, you don't want to expose them for longer because you kind of like, you increase on the clamping and also on the cell death of the Agrobacterium. Then after that, you decant and put the leaf segments with the Agrobacterium solution onto the cell strainer. You can use variable cell strainers. Others use an autoclavable mesh, as long as it is sterile. Then now you use paper towels, as you can see, and then you can now remove the excess Agrobacterium. So what happens is that this is also important. So after removing the excess Agrobacterium, now our infected leaf segments are ready to be taken on the co-cultivation plate. Now, one thing that you see that here is that on our co-cultivation plate, you're seeing that we have the white man pepper. Instead of putting them directly onto the co-cultivation plate, on co-cultivation media, we put them on this paper because there are so many and it will be laborious for you to like to move one by one because and then you're supposed to move this to the next, which is like resting media. So when you put them, it is recommendable to put at least 1 seedling per paper or 1.5 seedling per paper, and then you spread them using a loop or forceps. So then after that, you put them into the plastic box and you take them to 21 degrees for 1 day. Then after that, you remove this paper. Now you transfer the paper onto resting media for 7 to 11 days. Now, depending on the time that you have. So at 10 days, you should be able to see fluorescent transit expression is really good. Somatic embryogenesis has already begun and you can see. Then after that, you transfer them to selection media, which has your selection, agent or antibiotic. Then you can see that, but after 21 days, you visibly see the color, on the selection media. Now, after the 21 days, there comes another step, is that because you're using these morphogenic transcription factors, you have to excise them out. And the question that has been answered by Mercy, depending on the promoter that is driving CRE. So here, because we use ABA, we use RAB17 promoter that is driving ABA. So this is an important step, because if excision is not done well, you won't have very good regeneration. So we make our ABA media. When we make our ABA media, it has to be fresh. And when you make the ABA media, then you put the color, the individual color now. You do not transfer the whole filter paper. You pick individual color. You make sure that you have a very good contact. Do not over dip your color into the ABA media, because you will cause more stress. Then also, this step is variable. Some people do 3 days on ABA once. For us in our lab, we've optimized this step whereby we can do two times of ABA. We put them on ABA for 4 days, then we transfer them to maturation for 2 weeks. Again, we do a second round of ABA, and then we do another round of maturation, because excision is really important here. But then also, we've also optimized whereby you can do once ABA treatment for 7 days, and then you do 2 weeks of maturation in the dark. Then after, you take them to the light chamber. So the purpose here is there has to be a very good contact on the media. So the other step also, which is really important here, is that when you transfer them from maturation in the dark to maturation media in the light chamber, so based on our experience, we have seen that from a time of 5 to 7 days in light, it is really important, because when you begin to see green tissues coming out, the shoots beginning to form, it's important for you to transfer them to rooting media immediately, because if you don't do that, they will die off. And you can see the different days at 7 days, how your shoots are forming at 10 to 14 days. And then within 21 days, your plants, you've already seen your plants. So it's really important at every stage. So next, for people that have been working with sorghum, you know that actually different genotypes of sorghum have varying level of phenols. So in our protocol that was published by Massey, Massey was able to demonstrate that even though you have genotypes that produces high phenols, you can be able to have very good regeneration. And how do you do this? The way you do this is that you have to do what we call multiple subculturing. So this multiple subculturing, for example, instead of leaving them for 21 days on selection media, you divide them, you just put them on 10 days, you change the media to another media, and then you leave. Otherwise, if you don't do multiple subculturing, the phenolics will kind of like kill your color, and of course, you won't be able to get very good regeneration. So at this time, we will now take questions from the part of explant preparation to regeneration. Thank you.
Thank you. So this is a question from Young Quinn. Is there any morphological difference between embryogenic calli and non-embryogenic calli? Do you only move the embryogenic calli to the next stage media?
Yeah, so that is a very good question, actually. So yeah, so based on my experience, when I was beginning this training, so at first I used to move the embryogenic calli and the non-embryogenic calli because I was so optimistic. But then I realized that when you move the non-embryogenic calli, they do not regenerate into plants. So it's advisable to move only the embryogenic calli. But this comes with experience of kind of like doing so many experiments. I don't know if I've answered the question.
I think
Yes.
so. Typically, because it's having whorl and wavum, and it creates an environment that can regenerate very fast. Rather than, so you can observe if it is not transformed, they grow slower, and when it is transformed, it's growing vigorously. So that's how we track the somatic embryos and further. So that's a one point maybe, and we always have the visual marker like GS green or any other marker. So we can use that to take those. Yeah.
And also to add on to what they said, so sometimes you can actually see if your calli have like undergoing somatic embryogenesis based on the stage, like based on how they look like. So most of the somatic embryos seem to have multiple points, like they look more rough to view. But then when you have some calli which are not really going to regenerate. They just look smooth. It looks like they are trying to transition between leaf explants into calli, but then they are not fully, they've not fully formed hardy calli. So those ones, most likely and 100% of the time, they don't regenerate. So you'd rather prefer to have the ones which have a more rough appearance rather than like those that look smooth and wet and all that.
Thanks for that detail. So the next question is from Morgan. How many seedlings do you typically use per transformation?
Yeah, thanks for the question. That is a very good question. So in our lab here is that actually we vary. So we can blend between 5 to 20 depending on the number of explants. But even you can even blend using less. So depending on the quality of explant that you have, it varies. And then also if the number of explant is varying, also the times that you are blending also is going to vary. Thank you.
Yeah, and we'll give more answer to this in the next part. Yeah.
Okay, great. Thank you. And then last question. So are chimeric events observed among the transformed lines? If so, what strategies are employed to minimize their occurrence?
Chimeric, what in terms of, because we only screen in terms of the GS screen expression. What does specifically it mean? The
I'm not
final
sure,
output.
Alexandra, if you can expand on this question, then our speakers can maybe kind of answer your question more completely. So we'll wait to see if they follow up with that. And then there were a lot of questions for this section. So I'll ask one more question before we'll move on. So someone had a question about the filter paper. Does this not obstruct the energy derivation from the medium? Why is blending necessary while excision might be sufficient for Agrobacterium infection?
Yeah, so thanks so much. And those are like two questions in one. So
Yeah.
yeah, exactly. So I'll start with the blending because blending is your wounding, because actually when you look at the regeneration pathways in monocots, is that wounding is really important to activate other regenerative transcription factors. But even though you are inducing wounding, you don't want to do it so much because the kind of like the regeneration pathways and the wounding pathway is the same. It can lead to defense or it can lead to regeneration. So I would say that blending, you want to blend so that you can have uniform explants that is kind of like that can be induced to regenerate into plants. But at the same time, you don't want them to die off. So for the blending part, how many times, the type of blender, I think it's going to be in the next section and we'll answer more about that. So the other one is about the Whiteman paper about obstructing. So this protocol has been tested by our lab and it has been tested by whatever, if you can read the Wang et al. 2023 paper. So far, we are not seeing any kind of like differences. The only reason that's why we use the Whiteman paper is because if you are applying one leaf segment at a time, you are looking at the time and the labor cost and the efficiency, the transformation efficiency now versus the transformation frequency. So far, it's working out well. That's what I can say. Maybe my colleagues can add on that. Thank you.
Yeah, just to address the first question about why we do the blending, because so first of all, the goal is that you want to get as many regenerants from your transformation as possible. So you can decide not to blend your leaf explant, just cut them and put them in the agrobacterium. But let's say you are only going to get contact with agrobacterium from either side, like either cut ends of the explant, but when you chop them or when you blend them, it gives, because we are looking at multiple cellular regeneration, so that means you end up having multiple leaf pieces, which then end up having multiple exposure to agrobacterium. So that is why we blend. Of course, you can also hand cut, but I'll explain more on the blending as well in the next session. Yeah, and like Faisal said with the second question, if you have one leaf explant and then you chop it, you're going to have probably about 20 to 50 explants. So you don't want to just move all of them individually into each media, and then the following day you have to take your time and transfer each one of them. It just takes so much time. And the filter paper is thin, so the moment you put it on the media, you immediately see that it soaks up like the media. So it shouldn't really be much problem to getting all the nutrients out of the media.
All right, great. Thank you. So again, just a reminder, please put your questions in the Q&A box, not in the chat. It just is really, it's a lot easier to keep track of the questions. If we didn't get to your question, we will try to come back to it at the end. So with that, let's move on to our next section.
Okay, thank you very much. So this session is mainly just to address some common mistakes or some common problems or questions that we believe other researchers may have, and just to help in answering them. So the first one is what kind of blender do we use? So the specific blender we use is called Cuisinart's mini food processor, and then this is the specific details for it. Now, unfortunately, this manufacturer does not make this specific model anymore, but then they have one currently which is like quite similar to this. We have not tried it yet. We have not tried that. We still use this because we have a number of these that we are using. So you can get that and test. And then you can also try other models, because at the end of the day, the goal is to have a blender which has enough space for your explants to go around for easy processing. And then going back to the question that Morgan asked about how many explants do we blend, and Faisal answered that we blend at least about 5 or more. What we have even observed is that even in the case where you have about just 2 explants, so let's say you had a lot of seeds to sterilize, but then you did not end up getting all of them to be ready at 10 days, but you still want to carry out your transformation, we say that at least for even as low as 2 explants, please still go ahead and blend them. So there's an example of, we had just 2 seedlings after heat treatment, and then we chopped them, so you can see they look very small in the blender compared to the video that Faisal shared before. And then when you strain, this is how little it looks. And then moving them, it was like all on just one media. But when you compare this with hand cutting, now you can only cut too many times when you're using the manual hand cutting. And because there's so much pressure, because when you need to hand cut, first of all, you have to use forceps to hold the leaf explant before you try to use the blade to cut. It means there's so much manipulation and wounding, which is excessive, and that might not really help with the regeneration. But in the case of the blender, it helps to get multiple random cuts, which helps with a higher regeneration. So we suggest that even if you have very few explants, still go ahead and use the blender. It would just cost you a little bit extra, 700J, but for regeneration, that's a very minimal trade-off. And another question of low to no excision. So in this case, the construct we've stated the promoters that help them to be expressed. So we have NOS for Wushu, and then ubiquitin for baby boom. But then you can also check out other promoters. So if you read the Wang et al. 2023 paper, they tested different promoters and different arrangements on the tDNA construct. So you can use that as a starting point. But we've noticed that based on the promoter you are using to drive your morphogenic transcription factors, they can have a major effect on the regeneration. Now, if that is not the problem, and then we come back to the ABA step, which I believe Faisal has explained extensively, but the key points, try to have fresh ABA media or stock. We always make the ABA stock that we use, the 50 micromolar ABA stock, freshly. So you don't need to make so much. You can just make about 1 ml and then use within a week or a week, and then that's all. And then the ABA media itself, we just make enough to use for 1 or 2 experiments. If you have multiple experiments within a week, then you can make multiple plates. But if not, we just keep it to a minimum, and then we do 7 days at a go. The color might look a little bit stressed, but then we've realized that this actually improves the efficiency of regeneration, or you can do the 2 times ABA treatment. And so this is an example of a practical thing I faced in the early parts of the leaf transformation. So this genotype produces a lot of embryogenic calli. So trying just to do three days of ABA treatment, and then I realized that most of the regenerating shoots looked like this. So they had this coily, fatty roots. Some of them had the fatty roots, and these are the phenotypes that you see if the morphogenic transcription factors have not been fully excised. So from there, I started the whole experiment with just 15 explants, but then most of them had already moved to the ABA, 1 times ABA. So there were 12 plates in all, but then for 3 of those plates, I decided to do a second ABA treatment. And from a total of 9 plates, I only had 94 good plants. So like, for instance, those that are on this side, we had much less plants, but from just 3 plates that I did a double ABA treatment for, we had 47 plants. So imagine if I took all the plates to a 2 times ABA. So of course, if you do 1 times ABA, you would be lucky to get some regenerants, but then you just improve that better if you do a 2 times ABA treatment. And the other thing that's FISO address, which is at what stage should you move your regenerating shoots to rooting media? So like in this plate, the moment you start having the leafy shoots appear on your color, we advise that you move them immediately, like within 2 or 3 days, just so that they look green and nice at this stage, but shortly after that, they start to die off, and you don't want to lose your shoots after all the tissue culture. So this is a very important step as well. So thank you. And if there are any more questions, we would like to address them.
Thank you. So this is a question from Yang. How do you calculate the transformation efficiency? Is it based on the seedling number?
Okay, thank you for the question. So now we mentioned that we are experiencing the somatic embryogenesis per cell or per tissue. Now, on some calli pieces, there are multiple foci points of insertion. So like in the earlier slides where you saw the calli, there are like multiple points of which you see the Z has been being inserted. Now, if there are calli points, if there are regenerants which are growing in a cluster, we take them as they are coming together as one clonal regeneration. But if there are calli pieces that can easily separate and then there are divisions of calli junctions between them, you count those as independent events as they are not coming from the same calli point. So that's how we count those. So if you have multiple plants bunched together, we count all of that as one. But if you have multiple plants coming from different points of the calli or different leaf segments on the media, we count each of those as independent events and divide that by the number of explants you started with times 100, and that's the transformation frequency.
Great. Thanks for adding that detail. So we got multiple questions about the blender. A lot of questions about how do you sterilize the blender? Is there contamination with the blender? Could someone just maybe just add some more detail on how you keep things clean?
Yeah, on sterilizing the blender, so what we normally do is that actually before you do the explant preparation, so you get the blender, you use 70% ethanol, you kind of like wipe the blender, the blade and everything, then you let it dry. Then you kind of like you wash it again with autoclaved DI water twice. Then you let it dry. Then again you wash it again with 700 J before you use it for blending. Then after that, then you put in your 700 J. But then after doing the blending, so what we normally do is that because you want to avoid any kind of like contamination, is that after now you use I use like 50% bleach, like for 25 minutes, then I clean the blender. Then I let it dry.
Yeah, I think that's very important. Like as Faisal mentioned, like once you perform your experiment, is the 700J also having some nutrients, so it can. So don't keep the blender before. I mean, you should clean the blender after your H experiment. So clean it, dry it, and then reuse after sterilizing with ethanol and other like water. Yeah. we never, I mean, we hardly get any contaminants without doing even any autoclave, because these blenders are not autoclavable.
Yeah. And after the transformation, when you soak them with a bleach, please remember to clean them that same day, because when you leave them overnight, your blades rust and you don't want that. So it's best to clean them the same day after you are done with the transformation, and just to prolong the longevity.
So our next question is from Young Quinn. Is the humidity level in the incubator important at the callus induction stage?
So during callus induction, we do callus induction we don't need humidity after for the remaining. The most important thing is temperature. So that's what we try to control. But the humidity and all that is because of the heat that we expose the plants to before transformation. Yeah, for the remaining tissue culture steps, we don't need humidity for any step.
I guess have you seen a high humidity negatively impact the regeneration? Like, I'll just give an example of I've seen people try to keep their calli in growth chambers and then some people do it in the open air like racks, and sometimes you see a lot of condensation in the growth chambers.
Exactly. So I'll just say that actually, what you would want to prevent is too much humidity during your tissue culture process, because when you have too much humidity, that means that there's a possibility of having much droplets and all that, which might actually contaminate your explant. So that's also an important point in terms of when you make your media. You want to make sure that the media is well dried before you move your explant onto them, because you want to keep the condition as dry as possible, not extremely dry, but then dry enough so that you don't create a moist condition for any external contaminants to grow on them. Yeah.
Great, thanks for that clarification. This is a question from Adam. Is it possible to obtain the vectors from you? I assume you guys do AdGene, right?
Yeah, or
Yeah.
Corteva.
Okay, so AdGene or Corteva. All right, we have a lot of great questions. This was a question from Sandia. Are you using any supplements in the media to reduce phenolic compounds in the media?
Okay, so in the beginning, I've tested a few of what is available, like PVP, charcoal, what's it called? Yeah, activated charcoal. So I tried activated charcoal, PVP, and then coconut water. So coconut water actually helps with the proliferation of shoots from some of the recalcitrant lines. But then what I also saw with the use of PVP and activated charcoal is, even though they reduce the phenolic compound, they also kind of had an adverse effect on how much somatic embryos we had. So the explants did not look too happy. Of course, we still had plants, but they did not look too happy. So to help get as much as I wanted, I rather resolved to doing multiple transfers. So it's just the same as the other protocol, just that we just move them more frequently without any extra stress on them.
So we have another question here about the Agrobacterium maintenance. So this is, how do you transform and maintain the LBA 4404 thymine agrobacteria, and how do you transform those already harboring the PHP 71539 plasmid?
So for storage, we prepare our own in-house made competent cells, and we can transform with our electroporator. And usually we typically store in the glycerol in the form of glycerol stock, like roughly maybe 30 to 40% glycerol, and we try to maintain multiple glycerol stock in -80, so that we should not. So recently we came to that kind of information, like we should not rethaw the same glycerol stock several times. It can have so many problems, and because Agrobacteria itself can have so many modifications in their genome. So that's why we started to prepare the mother plates, and we can do multiple experiments in the same week or the next week, and we can have many experiments with that same one cycle of thawing. So try to maintain the glycerol stock at the freeze condition and without thawing completely. That makes life easy. I forgot the second question. Sorry.
Oh, how do you transform those already harboring the PHP 71539 plasmid?
I'm not sure about the maybe it's calling out the helper. So we just change the different antibiotic because maybe the backbone is kanamycin, and then if he's saying about the helper, maybe it's a streptomycin, so we have no problem with that.
Okay, great. Another question about Agrobacterium. How critical is it to use the REC-A deficient LBA 4404 strain?
Yeah, so this is also one critical aspect. Ideally, we also observe some recombination event when we transform our plasmids. So roughly, we observe like 10 to 20% there is some problem when you are using those. But yeah, I think that still so far it's not a serious problem. Yeah, but that's why we always check the DNA. Once we transform them, we isolate the DNA and retransform into E. coli and then check by using digestion or sequencing that is stable or not. So the first once we transform them, we need to verify or validate the strains that these are fine. You may have some recombination event or arrangement, so that need to be verified before going to the transformation. So that is a very critical point.
Great, thanks. So we have a question from Kelvin. Does the age of the explants have any negative impact on regeneration?
Yeah, so based on our experience and based on the protocol that we are using, yes, it does have exactly. So that's why it's important for you to use explants that are between 10 to 12 days. So the more you exceed the days, you may have some regenerants, but you reduce on the transformation frequency.
Thanks. And then everybody always asks that question for every protocol. Everybody wants to buy themselves more time, right? But the answer is always, unfortunately, you have a very short window. So our next question is from Todd Jones. So RAB17 is also inducible by dehydration or cold stress. Have you tested any other induction method other than ABA?
Wow, that's a very good question, and I'm happy that it's coming from Todd. So what we know is actually RAB17 can be induced by other abiotic stresses like cold or even warm temperature. That has been proved by literature. But however, the efficiency of induction, because I mean, even the cold stress and the warm stress is going to induce the ABA responsive pathways. Rather than us kind of like rely on other pathways that turn on the ABA, we just expose them to ABA directly. And this has been proved to be more efficient.
And additionally, the critical part is the leaky expression, because we know many of the individual promoters are not very specific to the inducer. It's also having a leaky expression, and that may end up of excising our Weibo mucil, and that we never want. So if we excise before it should be excised, we may end up of getting not any plants. So that's why I think Oliver already tested several different promoters, and RAB17 and heat inducible promoters, those looks very nice, and they have a very good regeneration efficiency. So that's why we are also doing the same way.
Great. Well, thank you so much, everyone, for all of your interesting questions. I think we generated some great discussion today. I hope that this workshop was really helpful to you. I learned a lot about sorghum presentation, so I just want to thank our speakers again for their time and their expertise and for sharing that with the plant gene community. We are at the end of the hour, so we will be ending. I know we weren't able to get to everyone's questions, but if you are a Plant Gene member, you can access our private discussion forum. So if you go to the website, all the information is there, and you can ask a question to the Plant Gene community. It's a great way to connect with other people, and we can discuss sorghum further. So again, just thank you all for joining today, and I look forward to seeing you at our next event. PlantGene is funded by the National Science Foundation. To learn more, please visit their website at nsf.gov.